Good afternoon. My name's Will Norton and I'm an Associate Professor of Animal Biology at the University of Leicester. The title of my talk today is Skin Swabbing is a refined technique to collect DNA from model fish species. In previous research published in 2017, my group and others had shown that skin swabbing is an acceptable technique to collect DNA from fish species used in the laboratory, such as the zebrafish or the stickleback. However, our original project, although it showed that we could collect enough DNA for PCR to identify animals, for example, didn't compare whether skin swabbing really was a refined technique that was less stressful to the animals being used than fin clipping, which is the standard procedure currently used in most laboratories worldwide. In order to investigate this, and with the aid of a research grant from the NC3Rs, we decided to compare 7 treatment groups in both zebrafish and stickleback. The first group were control undisturbed animals that were left in the tank. The second group were captured in a net and held underwater, whereas the third group were captured in a net and held in air briefly to simulate moving into another tank or being taken out to clip the fin or swab the skin. The fourth group of animals underwent the swabbing procedure in the absence of anaesthetic. In Group 5, we captured the fish and we anaesthetized them using MS-222, which is a standard aquatic anaesthetic for both zebrafish and sticklebacks. We held the animals in air and allowed them to recover. In Group 6, we anaesthetized the animals in MS-222 and swabbed them to collect DNA. And finally, in Group 7, we used a standard fin clip procedure in which we anaesthetized the animals and then fin clipped them, removing a small portion of the caudal fin with a scalpel. We performed 3 technical replicates and we used fish to look at release of the stress hormone cortisol, different behavioural and welfare indicators, which I'll explain in more detail later on in this talk, and qPCR to look at gene expression patterns. So our readouts were excreted cortisol into the water, behaviour and qPCR detection of gene expression. We decided to measure behaviour on days 1, 2 and 7. We first looked at the olfactory opercular beat rate and tail beat frequency as a measurement of stress or ventilation immediately after we performed the fin clip or the skin swab. 2 hours later, we then measured anxiety and locomotion using the novel tank diving, open field and black-white preference tests. We then repeated the novel tank diving, open field and black-white preference on days 2 and day 7 to look at medium-term effects of skin swabbing or fin clipping. We use qPCR to detect changes in genes which can be used as markers for the stress axis, and we also measured these on days 1, 2 and 7. We collected 4 brains from each treatment group and looked at brain-derived neurotrophic factor as a readout of neuroplasticity, and corticotropin-releasing hormone A and B, galanin and neuropeptide Y, all of which are part of the stress response machinery in the body. So by looking at these genes, we could see if the stress axis had been activated following any of these manipulations that we had already done. And finally, we looked at long-term changes to health after 1 month, and we used the health indicators, the hepatosomatic index, which can be used to look at energy storage, the splenosomatic index, which shows you part of the immune response, the gonadosomatic index, which is important for sexual development, and the pronephrosomatic index, which also shows part of the immune response. So this will tell us whether any of these techniques have long-term effects on the health and welfare of the animals. Our results were very interesting, but they're also a little bit complicated. And you remember we looked at 2 different species of fish, the stickleback and the zebrafish. And in all my graphs I depicted the stickleback data with black, and I depicted the zebrafish data using this brown coloration. On the top we can see the opercular beat rate per minute, and this is a readout of stress, so the animal is moving its operculum quickly and so ventilating very rapidly. We can see that in both zebrafish and stickleback, we see a change in the opercular beat rate when we use the anaesthetic MS-222. So that's groups 5, 6 and 7 on the right hand side of my graphs. Skin swabbing also caused a slight increase in opercular beat rate when we didn't use anaesthetic in zebrafish only. We saw a very striking result when we measured release of cortisol into the water. So this is the 3 unbound cortisol, which is excreted by the animals. and we saw that the only treatment which increased cortisol release in both species was group 7, so that's fin clipping, in both on the left hand side here sticklebacks and on the right hand side in zebrafish. And I've just highlighted here that in this data the most important groups to focus on are group 1, the undisturbed animals, group 4, which is skin swabbing without anaesthetic, and group 7, which is fin clipping with anaesthetic. So those are the standard treatments we'd use to collect DNA. We next went on to look at the behaviour data, and in general you remember we measured a large number of animals in 3 different replicates, and we combined these data to look at changes in behaviour, and we were surprised to see a very wide variation in the behavioural readouts that we used. So this data shows you time behaviour in a novel tank diving test depicted on the right hand side of my slide, and this is used as a readout of anxiety, and a more anxious animal will spend more time on the bottom of the tank, which is a thigmotaxis, a preference for the amount of time spent at the bottom of the tank. And the 3 graphs show you on the left hand side day 1 behaviour, in the middle day 2, and on the right hand side behaviour on day 7, 1 week after manipulation. Interestingly, in sticklebacks we only saw changes in behaviour on day 2 but not day 1 or 7, so these are medium term changes. And for example, group 4, which is skin swabbing without anaesthetic, swam slightly further than the undisturbed animals, group 1, and we saw a similar result for fin clipping, group 7. So it's intriguing to try and understand why we only saw a change on day 2 and not days 1 and 7. We also used the open field test, that's the top 3 graphs on day 1, 2 and 7 going across, and the black-white box, which is depicted on the second 2 rows of graphs and on the right hand side of my slide here. The open field test can be used to measure locomotion. You simply look at the distance swum in preference for side or middle of the tank, whereas the black-white test is a second readout of anxiety-like behaviour, and a more anxious fish will prefer the dark side and spend more time on the dark side of the tank. Once again, we saw very few significant changes in behaviour following any of these 7 manipulations in stickleback, so there are very few behavioural changes. So no significant changes in these behaviours in stickleback, regardless of the manipulation that we used. We used qPCR to look at the stress axis marker genes, and here we can see expression of both brain-derived neurotrophic factor and corticotropin-releasing hormone B in stickleback following fin clipping. So compare group 1 for BDNF, the top graphs, or CRHB on the bottom graphs, to group 4, and group 7 is fin clipping. You can see after fin clipping there's an increase in gene expression on day 1, which actually returns to basal levels over time, and we don't see a significant increase after skin swabbing without anaesthetic, that's group 4. We also saw that NPY expression was increased by fin clipping on day 1 as well as galanin. So here on the top we have no change in galanin, but NPY is expressed at a higher level. And that recovers over time, so on day 2 and 7 we see no significant changes in these behaviours. So we've already looked about stickleback. What about the second commonly used lab model organism, the zebrafish? Well, interestingly, we saw that zebrafish tended to show greater reactions in behaviour to these manipulations. They reacted more strongly than the stickleback. For example, if we look at the novel tank diving test, remember time at the bottom of the novel tank gives you a readout of anxiety, we see an increase of anxiety-like behaviour on day 2 when looking at group 4, which is the skin swabbing without anaesthetic, and that persists to day 7. So both in the distance swum and time spent at the bottom of the tank, although we don't see a difference on day 1. So once again we see these medium-term changes in behaviour. We also looked at the open field test for locomotion and the black-white test for anxiety. On the top graphs you can see distance swum in the open field on day 1, 2 and 7, and in the middle the time spent in the centre of the open field on day 1, 2 and 7. And here you can see a slight change in behaviour, a slight decrease in distance swum on day 1 following the fin clip procedure, and there's also a slight increase in the amount of time spent in the open field tank, which could indicate some changes in levels of anxiety. But that recovers over time, and we don't see these changes in behaviour on day 2 and 7. On the bottom row we can see time spent on the white side, so the aversive side of our black-white tank, and you can see while we do see a few changes in behaviour depending upon the manipulation group, so for example these animals in the first panel, group 2, were held underwater in a net, whereas group 6 is anaesthetized and skin swabbed. We don't see any changes in behavior caused by swabbing without anesthetic or by fin clipping with anesthetic. And finally, we did the same qPCR experiments in zebrafish, and we see very similar changes to those which we saw in the stickleback. So on day 1, we see an increase in the expression of BDNF, corticotropin-releasing hormone A, and corticotropin-releasing hormone B on the animals which have undergone the fin clipping procedure, so that's group 7. This persists to a small extent over time, so BDNF is still increasingly expressed more highly on day 2, but then by day 7 we see a recovery of the expression of all of these genes. And very intriguingly, we see a reduction of expression of corticotropin-releasing hormone B in the animals which undergo the skin swabbing procedure, so group 4, and we don't really know how to explain this decrease in gene expression. It's almost as if it's less stressful being skin swabbed compared to being unmanipulated, which is hard to believe. We also looked at changes in gadolin and neuropeptide Y. Neuropeptide Y, the bottom 3 graphs, we can see expression is increased by fin clipping on day 1, but that recovers over time, and we don't see any changes in the expression of galanin at any of the 3 time points we looked at. For the health and welfare indicators, we did see some significant differences in both species. So this is the data for stickleback. It's the data shown with the black data points. And we can see, for example, 1 month after manipulation, we see there's a slight decrease in body length for animals which have been given anesthetic, so groups 5, 6 or 7, and that also a slight decrease in weight. It's difficult to understand why this has occurred, so whether it really is a long-term effect of the anesthetic or whether just by random assortment we had fish of slightly different sizes in our treatment groups. And we saw very, very few changes in health and welfare indicators in zebrafish. Interestingly, the animals which underwent the skin swabbing procedure, so group 4, show a slight increase in body length, but only of several millimeters compared to the other groups. And again, this could be look at the draw in the way in which we assigned animals to different groups. So in summary, we compared skin swabbing and the standard technique used in the lab, fin clipping, to collect DNA, and we looked at different behavioral gene expression and release of the stress hormone cortisol, so these readouts at 30 minutes on day 1, 2 and 7 and 1 month. In general, we know that both techniques are suitable to collect DNA from model fish species. However, skin swabbing led to fewer changes in gene expression and behavior than fin clipping, although skin swabbing did cause some changes which were seen in the medium term, say days 2 and 7. When we looked at the spread of our data, particularly for behaviour, we were struck by the wide spread of data points. Some animals seem to not react to the DNA collection technique, whereas some react very strongly, and that suggests there could be intrinsic differences in some of the animals which we've looked at. We also saw that in some cases we saw a significant change in behaviour or gene expression on only day 1, so perhaps on day 2 or day 7 compared to day 1. And it's difficult to understand why this has occurred. It's possible that because we returned the animals to our facility in between taking these measurements across the week, that some other factor has influenced the expression of these readouts. For example, a change in the time in which they're fed, or the temperature, or the lighting conditions. It's very hard to factor in all of these different variables when you're doing such a long-term behaviour project which involves repeat measurements of the same animals. Strikingly, we saw that on day 1, fin clipping increased the release of cortisol, whereas skin swabbing did not. So that suggests that fin clipping is more stressful for the animal than the skin swab. And finally, we also noticed that fin clipping led to data that was much more variable than the data we collected after skin swabbing. And we tested this using the asymptotic test for coefficients of variance. And that's simply a statistical test which allows you to see whether changes in the spread of data are significantly different or not. And the upshot of this is that fin clipping will need you to use more animals in an experiment after the procedure. So you had to use a larger group of animals if you fin clip them compared to if you skin swab them if you want to detect significant results. So taken together, we believe that we have shown that skin swabbing is a refined technique to collect DNA from both zebrafish and sticklebacks because they show fewer changes in gene expression, behaviour and cortisol release. And it also has the potential to reduce the total number of animals that you use in your experiments because the data that you collect will be less variable. So this project was funded by the NC3Rs and we're hoping to publish it very soon. It was a large group effort and most of the work was done by Hector Carreño, who's on the right hand side of my slide here, and Kenwyn Tilley on the left hand side, 2 postdocs in the lab. And here you can see us in our lab skin swabbing zebrafish and looking very happy. Thank you very much for listening.